want to talk a little bit about breaking proteins down, because when we eat proteins and they are processed through our body through digestion, then we get the component pieces back. So those amino acids that make up the proteins, we get them back to reuse and put proteins back together. So in past videos, we've talked about putting together proteins through dehydration reactions. So we remove the water, we form these peptide bonds, and we can make these sequences of amino acids, which can then form proteins with different levels of structure. But if we were wanting to go the other direction, or if we wanted to talk about how did we get those component pieces in the first place, then we need to talk about hydrolysis. If you've seen some OCHEM before, then you know that hydrolysis reactions are pretty common, and they have to do with water coming in and breaking something apart. So lyse means to split apart, hydro of course referring to water. So this hydrolysis reaction is really the way that we get our amino acids that make up all of the proteins in our body. So it's an incredibly important reaction for the synthesis of all the proteins that do all of these amazing functions. So by definition, we have to add water to break apart some sort of chemical bond, and there have to be the right chemical conditions. So let's think about this with digestion. If we think about the first kind of steps of our digestive process, then we eat food, it goes into our mouth, it starts getting broken down immediately by our saliva, and it goes through our esophagus into our stomach. Our stomachs have high acidity to them, right? Our stomach acid is a pH of around 2-ish, and then food gets broken down there, and then it goes through our intestines, it gets broken down further and reabsorbed, and the different pieces and parts work, and then whatever we don't use or utilize then gets excreted as waste. So in that process, we needed something to break down the component pieces, and this all happens in the stomach. So this can happen under acidic conditions if we're talking about kind of this particular dehydration reaction. So it has to be in acidic conditions. So we can talk about this as kind of in the presence of hydronium ions or hydrogen ions. Or we could have it catalyzed by some sort of enzyme. Enzyme catalyzed. And there's a number of different digestive enzymes. So you usually hear about things like trypsin or chymotrypsin. These are enzymes that help to digest down proteins. They facilitate the hydrolysis reaction. And we'll get into enzymes and their functioning and how they work in future videos. But we know that enzymes are biological catalysts, so they speed up a chemical process. In this case, they allow for water to break apart these peptide bonds. So if we look at these, kind of here's a backbone of an amino acid structure. I've highlighted here the peptide bonds. And these peptide bonds are going to be the focus of the hydrolysis reaction, because then we're going to have water molecules that come in. So here's my bent water molecule. It's going to come in under these right conditions, and it's going to break apart this chemical bond. And then we might have a water over here that is going to come in and break apart this chemical bond. It's going to lyse it. And now we have this amino acid, but what happens with the water is the water also is going to separate. So the hydroxide piece is going to go on to the carboxylic acid and reform that carboxylic acid, and the hydrogen is going to go on the amine. So that's kind of a lot of arrows and messiness on my sheet here, but we can kind of also see it if we break out our handy dandy model. So here's my peptide bond. So I have a carbonyl group attached to a nitrogen, and if I have a water molecule that I bring in, so here's my water molecule, my hydrogen hydroxide, then this water molecule is going to come in and act on this particular bond. So if I break apart this bond, so I'm going to lyse the bond, and then I break the bond with my water here, then the hydroxide piece, the OH, is going to reattach to my carbonyl. So I've reformed my carboxylic acid here. So again, kind of carbonyl to the hydroxyl group that get reformed here. So that's half of my water. What's left over then is my hydrogen. And so if I go to the other piece of my molecule here, then this will reform my hydrogen on my nitrogen here. So now I have a full amine group, the NH2. and then this would be still attached to the other one. Then another water could come in and cleave the next one, right, which would be here, and then et cetera, and then you'd be left with kind of structures that are the amino acids on their own. Then they can do their own acid-base chemistry. You get those bitter ions, and then these amino acids can be used to be rebuilt into the proteins that are required. So we have this kind of back with the way that we had originally talked about amino acids when they were first introduced. And this is happening because the water is splitting apart that amide bond, the peptide bond, and half of the water goes to the carboxylic acid, the other half goes to the amine. So it's pretty straightforward. And again, if you've seen some organic chemistry before, it's probably not an unfamiliar reaction, but in the context of proteins, it's incredibly important because of this digestion process. So we need to break down these proteins. And if we think about protein in our diet, we're talking about meats, right, usually, or we're talking about tofu, or some of these other types of things that are high in protein. And these high in protein things we need to break down into their component amino acids, and then we can rebuild these amino acids into the proteins that are required to do the other functions in our bodies. So this hydrolysis reaction is very important, and if you have any questions on it, don't hesitate to reach out. Otherwise, I'll talk to you again soon.